Hi everyone, I'm XYXu from CSBL. The title of the journal that I'm going to introduce today is Surface Expression of Antitoxin on Engineered Bacteria Neutralizes Genotoxic Colibactin in the Gut, which is published on the Nature Microbiology. Here are the highlights of this paper. In this study, authors expressed intracellular CLBS on the surface of engineered bacteria to prevent the host from genotoxic effects across multiple PKS-positive isolates. The surface display effectively reduced colibactin-induced DNA damage and cell cycle arrest in human cell lines and organoids, outperforming diserin, a small molecule inhibitor of colibactin synthesis. The engineered strains mitigated intestinal damage in the mouse model of colitis and suppressed tumorigenesis in mouse models of colon cancer caused by PKS-positive E. coli. The results show the feasibility of inhibiting bacterial genotoxins in the gut, establishing a start point for therapeutics targeting other potential cancer-causing bacterial metabolites. Here is a brief introduction about the colibactin. Colibactin is a small molecule genotoxin made by certain gut bacteria that can chemically damage DNA and is linked to colorectal cancer development. And genotoxin is any agent that damages DNA or chromosomes and can thereby cause mutations, which may lead to cancer or heritable defects. And colibactin is encoded by the PKS, also called CLB, genomic island, and has recently emerged as a key microbial factor in the rise of colorectal cancer in younger adults. There are 19 genes from CLB8S are included in CLB island, and the gene introduced in this study is a self-resistance protein CLBS. Previously, the CLBS was identified as an antitoxin mechanism for PKS-positive bacteria to prevent colibactin-induced self-DNA damage. In this study, authors repurposed this toxin-antitoxin system using a microbial surface display platform to directly neutralize colibactin in complex environments, which is inspired by the bacterial surface display of whole cell biocatalysts in industrial application. And surface displayed CLBS completely blocks DNA damage from multiple PKS-positive strains in human and mouse colon cells and organoids. This approach suppressed intestinal DNA damage in the colons of wild-type mice and decreased tumorigenesis in a colitis-associated mouse CRC model by inhibiting the colonization and the procarcinogenicity of PKS-positive bacteria. To test this, authors sought to identify an optimal outer membrane protein scaffold to present CLBS on the bacterial surface. They screened 3 commonly used bacterial OMPs: lipoprotein fused with the truncated E. coli, outer membrane protein A, PPOMP-A, and C-terminal of IgA proteinase, and N-terminal of EAEA. And CLBS was efficiently displayed on the surface when fused to the C-terminus, such as OMP-A or MEAE, but not when fused to the N-terminus of CIJP. Additionally, diserin reported as a safe colibactin biosynthesis inhibitor, but in this study, the engineered E. coli OMP CLBS strain protects host cells from colibactin much more broadly and effectively than diserin. They also expressed OMPA alone as control strain to isolate the effect of CLBS display. Then co-culture these engineered strains with HeLa cells infected with E. coli DH10B host that harboring a bacterial artificial chromosome encoding the PKS island, or an empty vector that were previously developed by others. Gamma H2AX, the gamma H2A histone family member X, was used to assess the DNA damage by staining, a widely adopted method for detecting double-strand DNA breaks caused by PKS plus bacteria. And compared with this control strain, OMPA only and OMPA fused with the CLBS reduced the gamma H2AX signal by about 60%, indicating effectively neutralization of Calypectin-mediated genotoxicity. By contrast, MEAE-CLBS did not confer protection despite showing comparable surface display levels to OMP-CLBS, as shown in Figure B. So, they selected OMPA as the lead scaffold to characterize the surface displayed CLBS. Authors increased the plasmid copy number from a medium copy to high copy replication origin to enhance the surface display levels of CLBS and anti-Calypectin function. To test the anti-Calypectin function of surface displayed CLBS, they used two different strains express PKS gene to induce genotoxicity in hello cells. One is PKS positive NC101, which is known for its procarcinogenicity and adherent invasive nature in mice, and another one is DH10B harboring the PKS plasmid. And the results show that surface displayed CLBS can completely suppress the genotoxicity in both strains compared with the negative control. NC101 alone drives a strong G1 arrest in the hello cells, seen as a marked loss of 2N, which is diploid cells, compared with the NC101 PKS knockout control. When optimized OMPA-CLBS is present, this Calypectin-induced cell cycle arrest is essentially abolished, whereas OMPA without CLBS gives no protection. Together with the gamma H2AX data, this shows that the surface displayed CLBS not only lowers DNA damage but also prevents downstream cell cycle arrest in host cells infected with PKS positive bacteria. And 7 additional CLBS homologs, which 30 to 80% identity to E. coli CLBS, were codon optimized, put on the high copy plasmid, and expressed on the surface. Different homologs showed different levels of protection, but E. coli CLBS was still the most effective at reducing Calypectin-induced DNA damage. They also extended the surface display strategy to other E. coli chassis such as ECN, used for the delivery of biologics in preclinical models. Notably, while ECN has been used safely for over a century as a probiotic E. coli strain to treat intestinal disorders, the discovery of its genotoxic potential has raised recent safety concerns since it harbors the PKS island encoding for Calypectin biosynthesis, as well as another PKS-positive mouse E. coli isolate, NGF-1, were also used as control. The authors showed that ECN and NGF-1 are genotoxic in vitro due to their PKS islands, but expressing OMPA-CLBS on their surface abolishes this intrinsic genotoxicity while leaving PKS intact. They also co-cultured the different PKS-positive strains with NC101 models, and the results demonstrated that the bystander protection and the chassis performance in a more realistic mixed bacteria context. However, K12 strain was chosen as the preferred chassis for subsequent work because the highest protection ability was shown in K12 strain rather than in ECN, likely because ECN CLBS must neutralize both its own Calypectin and the NC101s, whereas K12 only deals with the NC101. To evaluate the antitoxic displaying approach in intestinal epithelial cells, authors established a co-culture model consisting of K12 E. coli strain with OMPA-CLBS, NC101, and the various CRC cell lines, using K12 E. coli strain with OMPA only and NC101 PKS knockout strain as negative controls. And the surface displayed CLBS in K12 strongly protects both CRC cell lines and the colon organoids from Calypectin-induced DNA damage and the cell cycle arrest. Next, they evaluated the efficacy of OMPS-CRBS in suppressing NC101-mediated host DNA damage in mice maintained under specific pathogen-free conditions. Mice were given streptomycin and DSS to inflame the gut, then chronically infected with NC101 or NC101 PKS knockout strain, while receiving repeated oral doses of OMPS-CRBS or OMPA only to test whether surface-displayed CRBS can reduce NC101-driven DNA damage in an inflamed intestinal environment. And the results showed that the orally delivered OMPA-CRBS safely reduces Calibactin-driven DNA damage and inflammation in the mouse colon, without detectable systematic toxicity or strong systematic immune responses. After validating the prevention from host DNA damage, they next examined whether this strategy could lower the tumorigenesis induced by PKS positive E. coli in colitis-associated mouse CRC model using DSS-treated APC mean plus mice. The authors show that orally delivered OMPS-CRBS significantly lowers Calibactin-driven colon tumor formation in the SS APC mean plus mice in a CRBS and PKS dependent manner, and that this effect generalizes beyond NC101. Because higher levels of the procarcinogenic NC101 correlated with the greater colon tumor burden in the SS APC mean plus mice, the authors next asked whether surface-displayed CRBS, which already prevents intestinal DNA damage and tumorigenesis by PKS positive E. coli, also reduces NC101 abundance in vivo. And this in vivo findings suggest that the engineered E. coli displaying CRBS not only suppresses Calibactin-mediated host DNA damage and tumorigenesis, but also reduces the abundance of procarcinogenic PKS positive NC101 in vivo. In this study, authors showed that surface-displayed CRBS can prevent DNA damage in human and mouse colon-derived cell lines and organoids, reduce tumorigenesis in the colitis-associated DSS APC mean plus CRC model, and selectively suppress PKS positive E. coli without affecting isogenic knockout strains in conventional mice. However, one potential limitation of this study is that the evaluation of Calibactin-induced genotoxicity has primarily relied on gamma H2AX staining and cell cycle analysis. While these assays are widely used and informative for assessing DNA damage and its cellular consequences, they do not capture the full spectrum of mutational events associated with Calibactin exposure. From the translational perspective, this study provides early proof of concept for a preventive strategy targeting early life exposure to Calibactin, a known contributor to mutagenesis decades before CRC becomes clinically detectable. From the fundamental perspective, findings on the targeted inhibition of PKS positive NC101 colonization by antitoxic display may reflect the complexity of the PKS island. This is the end of my presentation. Thank you for your listening.